Hello students, this is Geethanjali. In this video, we'll be learning about transformation. So transformation is a method of gene transfer in prokaryotic cells. That is the bacterial cells. This is a phenomenon that is most closely resembling with the sexual reproduction found in eukaryotes. Okay, so this process was actually performed as an experiment by Frederick Griffith in the 1928 year. Okay, so basically during those years, there was actually a hunt for the uh, genetic material to know what was actually the genetic material. So in 1928, Frederick Griffith, he used a bacteria called Streptococcus pneumonia, and he used two strains, smooth, shiny strain and one rough strain. Okay, so this smooth, shiny strain is actually shiny and smooth because it has a mucus coat, okay, made up of polysaccharide, whereas the rough one does not have. That's why we call it rough. So the smooth one is actually virulent, means it is pathogenic. It can cause disease, that is the pneumonia, whereas the rough cannot. So what he basically did is he first took the S strain and injected it into a mice, and he found out that the mice died because of pneumonia. And he took the R strain; it did not die because R strain is not capable of causing pneumonia. Later, he took a S strain, which was heat kill, which was subjected to high temperature and it was inactivated, so it couldn't cause the disease in the mice, and the mice still stayed alive. But next time what he did is he took a S strain heat killed bacteria and then R strain bacteria, which was live, and he injected both of them into a living mouse. Then he noticed that the mouse still died. So the only possible explanation for this is that something from this inactivated S or the heat killed S strain transferred to R strain, which made itself convert to a S strain. So the only difference we know of these two strains is that this one has a mucus sheath and this does not have, and this is virulent and this is non-virulent. So this was the only difference. So something would have been transferred, some gene responsible for these two characters would have been transferred to the R strain to convert it into S strain, right? So that was the only possible way to comprehend this. So they thought that this happened because of some transforming principle, because it actually transformed the alive R strain to a S strain. So few years later, they performed an experiment to find out the nature of this transforming principle, the biochemical nature of it. So Oswald Avery, Colin McLeod, and Maclean McCarthy, they performed an experiment. So they basically isolated the biomolecules that were usually suspected to be the genetic material, that is proteins, DNA, or RNA, from the S cells, that is the ones that cause the disease. Okay, and then they transferred them to the R bacteria. So in this case, what they found out was when they transferred DNA, transformation happened. Okay, so that means basically DNA is the genetic material, that is the one biomolecule compromising the genes. Then to confirm this, what they did is they took proteases, that means protein digesting enzyme. They took RNases, RNA digesting enzymes, and also they took DNases, DNA digesting enzymes. So when they used these two in the experiment together, they did not affect the transformation. Whereas when they used DNAs, transformation did not happen at all. So this suggested that DNA was the transforming principle. But many scientists were not convinced by this. So other experiments had to be performed to prove that DNA is actually the genetic material.